building on the discovery of the structure of DNA and learning a little bit more about the structure of DNA in itself, how exactly is it packaged together from chromatin to chromosome. So as you can see in the bottom left, that's a strand of DNA, a double helix. Again, every picture does look a little bit different. Towards the center of the picture, you see those are little proteins. Those are called histones. Histones are the protein that's responsible for getting DNA very condensed. And essentially, the DNA double helix is wrapped several times around the histone. When you have the histones bound together with DNA coiled around it, they form beads. Almost looks like beads on a string, as you can see towards the right-hand top side of the diagram. And histones together with DNA has another name, of course, called a nucleosome. So this is the same diagram from the last slide, but just a further picture beyond the histones wound up together with the DNA. So eventually you have tons of histones and DNA all coiled and looped together, and you have interactions of bonds keeping it all together, not all of which is understood by scientists. And then during all those phases, on the last slide up to this point, we're in interphase. So we have to remember of the cell cycle, interphase is where the DNA isn't getting ready to divide until mitosis is signaled or starts to begin. So when you see on the right-hand side that chromosome that has been, hopefully you can tell, it kind of looks like an X a little bit. You can see that it's now been replicated, indicating that mitosis is most likely going to take place. And when that happens, we call it a condensed chromosome, or, because now it's been copied, two chromatids bound together. So we have a summary of how DNA can vary from a prokaryotic cell, like bacteria, versus a eukaryotic cell, such as animals and plants have. So first, prokaryotic DNA is often double-stranded, or is always, double-stranded, the same for our DNA as well. Prokaryotic DNA is circular, and our DNA is linear. Prokaryotic DNA is, because it's a single-celled organism, there's not too much room, so often you have one chromosome, and in eukaryotic cells, you have one, possibly like in a yeast cell, or many chromosomes like ours. Prokaryotic DNA, for where is it located? It's in the cytoplasm or the cytosol of the cell, and then our DNA is located in our nucleus. Now, when prokaryotic DNA is in the cell, it is supercoiled together, and we usually call that a nucleoid. In eukaryotic, as I just explained on the last two slides, you have chromatin if the cell is in interphase, and then you have histones, which again are proteins that the DNA is wrapped around. Now, one thing I forgot to mention on a slide or two ago is the reason the histone is attached to that DNA has to do with the charges. So DNA, the outside, try to remember, because of those phosphates, it has a negative charge, which indicates that the proteins have a positive charge.